The promise of genetic medicine is pretty staggering, isn't it? I mean, the idea of actually fixing diseases right at their source, whether it's something inherited or even taking on something as complex as cancer.
It's a powerful idea, yeah.
And it seems like, if you're following this stuff, you see the buzz about all these clinical trials. There are thousands of them. But then you look at how many actually get approved, and it feels like we're kind of hitting a wall.
It does feel that way sometimes, huh?
Like these therapies, they face real challenges with how our bodies deal with them, how easy they are to actually manufacture on a large scale. And then there's that whole thing about our immune system sometimes seeing them as a threat.
Yeah, you hit the nail on the head there. The idea is incredibly powerful, but the delivery, the actual getting those therapeutic molecules to where they need to go safely and effectively has been a major hurdle. Tolerability, scalability, immunogenicity, these are really the key challenges that researchers are grappling with.
Which brings us to the heart of our deep dive today. You shared this really fascinating article from Cell. It came out fairly recently, September 2024.
That's right. Yep.
Titled Safe and Effective in Vivo Delivery of DNA and RNA Using Proteolipid Vehicles by Brown and colleagues.
A great paper, really groundbreaking stuff.
Yeah, it really seems like it is. So this article introduces proteolipid vehicles, or PLVs, as a novel way to deliver DNA and RNA.
Right.
Now, this isn't just another viral vector or your standard nanoparticle, is it?
Not quite, no. It's a kind of a hybrid approach, you could say.
It sounds like it's kind of taken the best of both worlds.
Precisely. It takes advantage of what we've learned from lipid nanoparticles, which have shown good success, especially with RNA delivery. Think of the recent mRNA vaccines.
Right.
And it combines that with something really interesting borrowed from viruses.
Okay, so what's the viral piece of this puzzle? The article mentions these FAST proteins. They come from fusogenic orthoroviruses, which, for our listeners out there, those are actually a family of pretty common viruses.
Right.
But that sounds kind of intense, right? Fusogenic orthoroviruses.
Well, it is quite descriptive. FAST actually stands for Fusion-Associated Small Transmembrane Protein. And the remarkable thing about these FAST proteins is that they are the smallest known viral fusogens. We're talking only about 100 to 200 amino acids in length.
Wow, so tiny.
Now, orthoroviruses, they're non-enveloped viruses. They're known for causing cell membrane fusion. So these FAST proteins are displayed on the surface of infected cells, and they essentially help the virus spread by causing these infected cells to fuse with their neighbors, forming these large, multinucleated cells that are called syncytia.
So they're basically making cells merge together.
Yeah, you could think of it that way.
So, these FAST proteins naturally cause cell membranes to fuse. How does that translate into delivering genetic material?
That's a great question. And the article highlights this really important point, that they do this at the body's normal pH. They don't need to bind to specific receptors on the cell surface, which is a huge advantage.
So why is that such a big deal?
Well, most current non-viral delivery systems, like your conventional lipid nanoparticles, they mainly rely on a process called endocytosis to get into the cell. So the nanoparticle gets taken up into a little compartment within the cell called an endosome.
Right.
And then it needs to break out of that endosome to release its cargo into the cytoplasm where it can actually do its job. But this endosomal escape, it can be really inefficient. It's often a major bottleneck.
So it's kind of like getting stuck in customs, right?
Yeah, exactly. Good analogy.
Yeah.
But FAST proteins, because of their unique ability to directly promote fusion with the cell membrane, they bypass that whole endocytosis step. They can just fuse directly with the cell membrane at the physiological pH of our body.
So they're like a VIP lane straight into the cell, basically.
Precisely. And because they don't need specific receptors to latch onto, they can potentially interact with a much broader range of cell types. So it opens up lots of possibilities.
That makes a lot of sense. So the article goes on to describe how they engineered a chimeric FAST protein called P14 endo-15. It sounds like they basically took pieces from different FAST proteins.
Yes, P14 and P15, to be precise.
To create something even more effective. What was the logic behind that?
Well, they looked closely at the different functional domains of P14 and P15. Things like the part that interacts with the lipid membrane, called the ectodomain and transmembrane domain, and then the part that's inside the cell, called the endodomain.
Okay.
Their thinking was, if we could carefully select and combine these modules, could we engineer a hybrid protein with even better membrane fusion capabilities?
So it's almost like they were doing like a mix and match, seeing what works best.
In a way, yes. And through a lot of testing and screening, they identified this P14 endo-15, which combines the exterior and membrane-spanning regions of P14 with the interior region of P15. And this particular combination turned out to have significantly higher and faster cell fusion activity in the lab compared to either of the parent proteins.
So they basically supercharged it. They found their superfusogen.
That's a great way to put it. And this P14 endo-15 is the key ingredient in these FAST-PLVs.
So how are these PLVs actually put together? We've got the FAST protein, but what about the proteolipid vehicle part? What else is in these delivery packages?
Well, at their core, PLVs are lipid nanoparticles. So think of them as tiny spheres made up of fat-like molecules.
Okay.
But with this engineered P14 endo-15 protein embedded in their membrane.
Nice.
The researchers really optimized the lipid composition to make sure it would be well tolerated by cells. They experimented with different types of lipids, including cationic and ionizable lipids, which help package the negatively charged nucleic acids, and then also helper lipids like DOPE and pegylated lipids like DMG-PEG, which contribute to the stability and prevent them from clumping together. And through this process, they arrived at a formulation that was not only super effective at encapsulating DNA and RNA, but also very gentle on cells.
So it's not just throwing things together, it's a very specific recipe.
Absolutely, it's very precise. They identified a specific formulation called 41N, which really hit that sweet spot between delivery efficiency and low toxicity.
And they actually confirmed that these PLVs were forming these spherical structures and that they were all consistently sized.
Yes, that's really important for how they behave in the body.
Right.
Using fancy imaging techniques like transmission electron microscopy and atomic force microscopy, they were able to actually visualize these fast PLVs and confirm that they were indeed forming these uniform spherical nanoparticles, typically less than 80 nanometers in diameter.
Tiny.
Very tiny. And this small, consistent size is crucial for them to be able to circulate effectively in the bloodstream and reach those various target tissues.
Okay, so we have these optimized fast PLVs loaded with either mRNA or DNA, but do they actually work? The article presents a lot of in vitro data, experiments done in cell cultures. What were some of the key findings from those initial studies?
Those initial studies were very encouraging. They showed that these fast PLVs could efficiently deliver both mRNA and DNA into a wide range of cell types.
Yeah.
And this led to significant production of the proteins that were encoded by that delivered genetic material.
So they were actually making the stuff they were supposed to make.
Exactly. Importantly, they found that having that fast protein really enhanced the delivery efficiency compared to similar lipid formulations that lacked it.
So it was really key to making the whole thing work.
It was. And despite the fact that the fast protein is so good at fusing membranes, they didn't see any significant unwanted cell fusion or syncytia formation at the doses they were using for gene delivery, which is a big plus for safety.
You don't want your delivery system going rogue and merging all your cells together, right?
Exactly. That would be a problem.
The article also suggests that fast PLVs might get into cells through a different route than the typical LNPs. They mentioned something about being largely independent of endocytosis.
Right. That's an important distinction. So, as we were talking about earlier, conventional LNPs, they mainly rely on endocytosis, where the cell membrane basically engulfs the nanoparticle.
Right, like Pac-Man.
Haha, yeah, like that.
Huh.
But the researchers did several experiments to show that fast PLVs, because of their fusion capabilities, can actually deliver their cargo directly into the cell's cytoplasm, basically bypassing that whole endocytosis pathway.
Cutting to the chase.
Yeah, exactly. They used inhibitors that block endocytosis, and they tracked where the delivered siRNA ended up.
siRNA being that small piece of RNA that can silence genes, right?
Yes, that's right. And what they found was that the fast PLVs led to a much more widespread distribution within the cell, and they didn't co-localize as much with those early endosomes compared to MT3 LNPs, which are a common type of LNP.
Interesting.
So this endosomal independence could mean more efficient and reliable delivery of that therapeutic genetic material.
But of course, the real test is how these things perform in a living organism.
That's the ultimate test.
Right. And the article describes some studies in mice and even non-human primates.
Right.
So what did they find out about safety and how well they worked in vivo?
The in vivo studies were really encouraging. They found that in mice, fast PLVs had a much better safety profile than MC3 LNPs. At higher doses, the MC3 LNPs caused a lot of liver toxicity and even some deaths.
That's not good.
No, not at all. But the fast PLVs were well tolerated even at much higher doses. They also look at the cytokine responses, which basically tell you how much the immune system is being activated.
And that can be a sign of toxicity too, right?
Yes. And they found that even at high doses, the FAST-PLVs didn't trigger the same level of pro-inflammatory cytokines that they saw with the MC3-LNPs.
So that low immunogenicity seems to be a really big deal.
It is. It's a major advantage.
What about how effective they were at actually delivering the genetic cargo to the animals? Where did the mRNA and DNA end up, and was it actually being expressed?
They saw a really broad tissue distribution with the FAST-PLVs. And this is a key finding. Many existing non-viral delivery systems, they tend to accumulate mostly in the liver.
Right.
But the FAST-PLVs were able to successfully deliver both mRNA and DNA to a much wider range of tissues in the mice. Importantly, the lungs, the spleen, the brain, the heart, and the kidneys. And they used luciferase as a reporter gene.
Which basically means its activity is easy to measure, right?
Exactly. It's like a beacon. And they saw a significant gene expression in these tissues outside the liver, in some cases much higher than what they achieved with MC3-LNPs. And they even showed sustained DNA expression for up to a year in mice after just a single systemic administration.
A year?
Yes, which is pretty remarkable for a non-viral vector.
That is really impressive. And this wasn't just in mice, they also looked at non-human primates.
That's right, which is obviously a much closer model to humans.
Right, definitely.
And those studies really backed up the safety and broad biodistribution findings that they saw in the mice. They gave the primates FAST-PLVs carrying a non-coding DNA sequence, and they found significant levels of that DNA in all 30 tissues they looked at.
Wow, all 30.
With particularly high levels in the lungs, spleen, gallbladder, and bone marrow. And notably, there was greater uptake in these organs compared to the liver, which is kind of the opposite of what you usually see with these systems. And in terms of safety, they didn't see any significant abnormalities in any of the organs. There were only some mild temporary elevations in some clinical chemistry markers and cytokines right after the infusion, but those quickly went back to normal.
That's good.
Yeah, it is. And importantly, they didn't see a strong induction of antibodies against that FAST protein in the primates either.
Again, with the low immunogenicity, this seems to be a real recurring theme here.
It really is. And it has huge implications, especially for the possibility of repeat dosing.
Because that's one of the big challenges with most viral vectors, like AAV, right?
Exactly.
Right.
After that initial dose, the immune system kind of learns to recognize them, and it becomes harder to use them again.
So what did this study show about redosing with FAST-PLVs?
This is another area where things look really promising. They did some repeat dosing studies in mice, where they gave FAST-PLVs carrying the luciferase gene either into the muscle or intravenously once a month for 6 months.
Wow, 6 months.
And what they found was that they got consistent reported gene expression after each dose with no significant decrease in activity over time.
So it kept working.
It did. And really importantly, they found very low levels of antibodies against the FAST protein in most of these repeatedly dosed animals. And even in the few cases where they did detect antibodies, those antibodies didn't show any significant neutralizing activity in their lab tests.
So they weren't interfering with the delivery.
Right. Which strongly suggests that you could potentially use these FAST-PLVs for gene therapies that need multiple doses, which is a big limitation of current viral vectors.
So we've got a good safety profile, broad delivery capabilities, but what about the actual therapeutic potential? Did they show that these FAST-PLVs could actually treat a disease?
They did, yes. To demonstrate the therapeutic potential, they focused on delivering a gene for follistatin, which is often shortened to FST.
Okay.
Follistatin is a protein that can promote muscle growth by blocking the activity of myostatin, which is a natural inhibitor of muscle development. It's actually being explored as a potential treatment for muscle wasting disorders. So what they did was they delivered the follistatin gene, specifically the FST344 variant, using FAST-PLVs in mice. They observed increased levels of follistatin protein in the bloodstream, and importantly, these mice showed significant increases in both muscle mass and grip strength compared to the control animals.
So they actually got bigger and stronger.
They did, and these effects were comparable to what has been seen with AAV-based follistatin therapies in preclinical studies.
So that's a really promising sign.
It is.
So to summarize, these FAST-PLVs seem to be a pretty revolutionary new platform for delivering genetic medicines. They combine a well-tolerated lipid nanoparticle with that specially engineered viral fusion protein, P14 endo 15.
Really elegant design.
And the advantages that they saw in this study, they're pretty remarkable. We've got effective delivery of both mRNA and pDNA.
And not just in cells in a dish, but in living animals.
Right, both in vitro and in vivo. And that delivery goes way beyond the liver.
Yes, a big advantage over so many existing delivery methods.
We saw good results in the lungs, spleen, brain, heart, kidneys, all these places that have been hard to target.
And they saw substantial uptake in gene expression in those tissues.
Right. And then there's that low immunogenicity.
Potentially a game changer for repeat dosing.
Right, because you're not triggering a huge immune response against the delivery vehicle itself. And that's a big hurdle with those viral vectors.
You mentioned it earlier, but they didn't see significant neutralizing antibodies against the FAST protein, even after multiple doses.
Right. And they also showed that it's safer and better tolerated than at least one of the commonly used LNP formulations out there.
Those MC3 LNPs, yeah.
Which were showing liver toxicity and all those elevated cytokine levels at lower doses than the FAST-PLVs.
A big difference.
And then there's the versatility of how these can be administered. You've got systemic delivery, but then also intramuscular, intrathecal into the spinal fluid, intracerebral ventricular directly into the brain, even intravitreal into the eye.
Right, with localized expression depending on the route.
And finally, they showed that it's not just about delivering reporter genes. They showed real therapeutic potential with that follistatin gene, leading to increased muscle mass and strength in the mice, comparable to what they're seeing with those AAV-based therapies.
Very impressive results overall.
So taking all of this together, this flexibility, the safety profile, the ability to deliver both DNA and RNA to all these different tissues, including those that have been so hard to reach, it really makes you wonder, how might this platform change the game for treating diseases that have been particularly challenging? Things like those disseminated cancers that need widespread delivery, or conditions where you need very localized gene therapy in delicate organs like the brain or the lungs.
So many possibilities.
It really seems like FAST-PLVs could be a big step towards more personalized medicine and maybe, in the not-too-distant future, reducible gene therapies.
It's definitely something to watch closely.
For sure. A lot to be excited about in this field.